All right, good afternoon, everyone. Welcome to Department of Medicine Grand Rounds. So before I get started welcoming today's speaker, I just wanted to share the lineup for the next few weeks. So next week, we welcome Dr. Shailendra Bazan from Harvard University, who will present Optimizing the Diagnosis and Treatment of Hypogonadism. And then the next week is the Michael John lectureship guest, Dr. Christine Castle, who will present Academic Freedom, Constructive Dialogue, and Flourishing in Medicine. We need to talk. So today I have the pleasure of introducing Dr. Kara Mould, who is an associate professor here in pulmonology and critical care medicine, who I've had the pleasure of knowing actually for a while. We crossed paths at Emory when she was chief resident during my residency. So she received her MD and MPH from Northwestern before going to Emory for her internal medicine residency, where she spent a chief resident year prior to coming here for her fellowship, and then stayed on faculty here and is at National Jewish Health. Her research focuses on innate immune response and lung inflammation, particularly the transcriptional and metabolic programming of alveolar macrophages. And she's had support from multiple NIH R01 grants for her work and published in multiple leading journals. She has been an invited speaker at many national conferences and received numerous awards for her research, including as the American Society for Clinical Investigations Young Physician Scientist Award and the Rising Star Award from the American Thoracic Society. She also serves as one of our associate program directors for research in pulmonology and critical care. So please join me in welcoming Dr. Mould, who will be presenting today's Grand Rounds, Deciphering the Innate Immune Response to Lung Inflammation.
Thank you very much for that kind introduction. It's nice to see you again. I'm going to talk today about a project that started way back when I was a fellow and was both delayed and significantly impacted by the COVID pandemic, and is just now getting wrapped up, and this week will be submitted for publication. So for multiple reasons, I'm very happy to be sharing this work with you all today. In the longstanding tradition of Medicine Grand Rounds, I thought I would start with a case. And in this, for this talk, I'd like to compare two cases. The first, patient A, is a 59-year-old with diabetes who's admitted to the MICU with dyspnea and is diagnosed with influenza A. The patient's admission chest X-ray looks like this: bilateral, fluffy alveolar infiltrates, and they are treated with usual therapies and placed on heated high flow for about 6 days. They eventually go to the floor and are discharged home on oxygen with a plan to follow up with their primary care doctor and with pulmonary in a few months. They had a CT-PE CT when they were in the emergency room, and that didn't show pulmonary embolism, but did show these bilateral infiltrates in kind of all the lung regions, some ground glass, some dense opacities, pretty typical of what you'd expect for influenza pneumonia. Patient B sounds a lot like patient A. 55-year-old with diabetes and hypertension, came to the emergency room complaining of dyspnea, also had a CT scan that looks a lot like patient A with bilateral infiltrates, also has influenza, and is on heated high flow for about a week in the MICU before transferring to general medicine and being discharged home with oxygen, with a plan to follow up with a primary care doctor in a couple of weeks. And so as a pulmonologist, we see these patients in clinic. Patient A comes in and says, "I feel great. My primary care doctor told me I don't need oxygen anymore. I live in Colorado, so I'm getting ready to climb another 14er. Everything's great. I'm back to normal. I'm glad that I survived influenza pneumonia with really no side effects. Do I need to come and see you again?" And you say, "You know, your repeat CT scan looks pretty good. Most of these infiltrates have resolved. There are some small areas that look like they could have some scarring, but overall your pulmonary function looks good, and we're glad that you're feeling so great. You don't need to come back and see us." Patient B, though, feels terrible, is still on oxygen, is still dyspneic with minor everyday activities, Pulmonary function tests are restrictive, and their repeat CT scan shows persistence of many of these infiltrates and evidence of scarring. And the radiologist says this looks like fibrosis. And so the question that really informs the research I'm going to present today is, how do these two patients who demographic-wise looks the same, who both had influenza, and who were treated with the same therapies and had the same clinical course, have such varied outcomes? In other words, what are the molecular or biological drivers of their lung repair? And the hypothesis that we've been testing for many years is that it's the macrophage in the lung. More specifically, we think there are unique subsets of macrophages that may drive healthy versus fibrotic repair. So why are we focused on the macrophage? Well, it's true that I trained in a macrophage lab, and so I drank the Kool-Aid, but there's pretty good data to suggest that macrophages are at the center of the repair process in the lung. So many of us, when we think about the alveolar macrophage, have some version of this picture in our mind, where we draw a simple alveolar structure with the fragile type 1 epithelial cells lining the alveolus, and occasionally a type 2 cell. And those cells we know are very delicate and very thin to allow gas exchange to this pulmonary capillary that's nearby. And we usually draw a macrophage right in the middle, because we know that macrophages are important for recycling surfactant and for acting as sentinels of the innate immune response, such that if we inhale a pathogen or some other inflammatory mediator, these macrophages sound the alarm, and they call in the neutrophils to help control the infection. Those neutrophils are a little messy, and they cause capillary leak, and they might disrupt these delicate type 1 epithelial cells, and so then we get leakage of proteinaceous edema fluid into the lung, and now our alveoli can't participate in gas exchange. And so we think of these macrophages as being inflammatory, important for pathogen killing, important for recruitment of other inflammatory cells, and as a link between the innate and adaptive immune response through their antigen-presenting capabilities. But our research has really been focused on other functions of the macrophage, and those are the reparative functions. And so it's true that macrophages do all these inflammatory things, but once inflammation has ceased, once they stop calling in the neutrophils, they're also responsible for cleaning up all of the cellular debris. As the name suggests, they're phagocytes, and so they're important for cellular clearance and prevention of secondary necrosis of dead cells and persistent inflammation. They also secrete reparative growth factors and inflammatory, non-inflammatory mediators that tell epithelial cells and other cell types to repair. And it's thought that if this process goes unchecked, that this may lead to fibrosis. And so the focus of our group for many years now has been, how can this one cell type, the macrophage, do all of these different things? What is the switch that changes a pro-inflammatory macrophage to a reparative macrophage, and how can we make sure that those processes are in check? Well, we think that perhaps there are different subsets of macrophages. In this time of single cell technologies, we asked, perhaps there's some division of labor. There's a big literature about how microenvironmental factors may influence macrophages, but maybe there are many macrophage cell types that have to work together for these functions. And so again, if we think about a lung at rest, we think about these resident alveolar macrophages, abbreviated here RAM, and we know that, at least in rodent models, these macrophages are in the lung since birth. So they're embryonically derived, and they proliferate in situ and persist in the lung throughout the lifespan of an animal if it's unperturbed. If an infection or an inflammatory molecule makes its way down the airway and into the alveolus, this inflammatory cascade starts, and some of these resident alveolar macrophages die, either passively or in order to control the infection. And they're replaced by monocyte-derived cells that come into the airspace and can mature into macrophages over time, such that once inflammation has ceased, your macrophage pool is now heterogeneous. You have resident airspace macrophages that are of a different embryologic origin than those that are derived from monocytes. When I joined my mentor Bill Jansen's lab during fellowship, he had done work to show that these two types of macrophages could be identified and separated in the mouse model of lung inflammation. So he used a LPS, or bacterial endotoxin model of lung inflammation, and showed that if you give LPS intratracheally to a mouse, you get this very reliable inflammatory timeline, where by day 3, neutrophils peak in number. Once the LPS is neutralized, they undergo apoptosis and are removed from the air spaces by day 6. And at the same time, macrophages increase in number, and they stay in the airspace during this reparative phase when the lung is returning to normal structure and function. Using a number of bone marrow chimera studies and shielded radiation experiments, he showed that these two cell types could be differentiated based on their cell surface markers. And so this was a great setup for a fellow project where I took these two cell types at all of these different time points and performed bulk RNA sequencing to look at what the functions of these two cell types were. Was one pro-inflammatory and one more reparative, as we hypothesized, or was there division of labor between these two cell types? And that data is summarized by this heat map, which shows the global transcriptional similarity or dissimilarity of all these cells at all these different time points. And what you can see is in the upper right corner, resident alveolar macrophages at all these different time points, whether during health, inflammation, or repair, are all very similarly programmed to each other, whereas the recruited macrophage programming is very dynamic. So at day 3, those cells are very unique in the bottom left-hand corner. And then during reparative time points, the recruited macrophages are, again, very transcriptionally unique. And this was quite surprising at the time, because again, we really thought that macrophages were programmed primarily by their microenvironment, meaning if you're in the inflamed alveolus for a week or a week and a half, probably your behavior is going to be similar if you're a macrophage. And what this study showed is that the main driver of macrophage programming is really where it started, the bone marrow versus the lung. We went on to do a number of differential gene expression analyses and pathway analyses, and what we showed is that the genes that separated these two cell types were likely to ascribe unique functions, whereas the resident macrophages proliferate and really do homeostatic macrophage functions. These recruited cells are very dynamic. During peak inflammation, they make all of the inflammatory cytokines that we've associated with ARDS and lung inflammation, IL-6 and IL-1. They have unique metabolic programming, which we know is very closely linked to their immune programming. And then during the reparative phase, these recruited macrophages are also making most of these reparative factors that we think are important for fibroblasts and epithelial cells and all the other mesenchymal cells to repair the lung. And so this was very exciting to us, and we were very interested to know if these cells might also be responsible for fibrosis. In the lab at the same time as I was working on this project, my now colleague, who was then a postdoc in Dr. Jansen's lab, was directly testing whether recruited macrophages contributed to fibrosis. So Ali McCubbery developed a model of to test whether recruited macrophages could protect a mouse from bleomycin-induced fibrosis. She induced apoptosis in all of the recruited macrophages and showed that fibrosis was attenuated when recruited macrophages were removed. So in the upper left corner, you can see a gross section, a lung on both sides and a heart in the middle of a mouse with pulmonary fibrosis from bleomycin, and that's blown up on the bottom where you can see the thickened alveolar septae and infiltrates that we see in fibrosis. And again, if you remove the recruited macrophages and induce their apoptosis, that lung is protected. Since this time, and kind of at the same time, many other groups showed a similar thing. A group at Northwestern used basically the same setup, but targeted a different molecule, in this case, caspase-8, and showed that if you deplete recruited macrophages, a mouse can be protected from fibrosis. This has been shown in many other models of lung fibrosis as well. And so taken together, the overarching hypothesis at this time was that recruited macrophages might predispose to fibrosis or even cause fibrosis after lung injury. And there was this paradigm where we thought maybe whether or not you have recruited macrophages in your lung can determine whether or not you go on to dysregulated repair. And I think that's shown in this pictograph, again, emphasizing that When we're born, we have all fetally derived or tissue resident macrophages in our lungs. Then we meet our parents and we go to daycare and we encounter viruses and bacteria, and some of those tissue resident macrophages are replaced by monocyte-derived cells. As we go through adolescence and adulthood, we come into contact with environmental exposures, some of which we can't control and some of which are self-induced perhaps. And by the time we are old, we have primarily monocyte-derived macrophages in our lung. And perhaps that's why these folks are more prone to fibrosis. And so we thought this was a really compelling hypothesis and had important translational considerations. But before we could think about how this might affect people's response to an inflammatory insult, we thought we should understand the variation in macrophages in a cross-sectional cohort of people. And so we designed an experiment to just do bronchoscopy on some healthy adults and use single-cell RNA sequencing, which was quite new at the time, to understand the heterogeneity of macrophages in all of us here. And so that's what we did. We enrolled 10 people who were healthy as judged by a comprehensive physical exam and spirometry and EKGs. And we had representation across 3 decades of life, male and female sex, 10 points of BMI, and many different racial and ethnic backgrounds. We performed BAL and got good return, and we had plenty of cells by which to perform single-cell RNA sequencing. I worked closely with Dr. Camille Moore, who is a biostatistician at National Jewish, and she really taught me how to wrangle single-cell RNA sequencing data and how to interpret our findings. And what we found is if you look at this UMAP plot, where each dot represents the global transcription of a single cell, you see that we identified many subsets of airspace macrophages. It turns out that these are all resident macrophages, and although they have some slight differences between them, they would all be considered a resident macrophage. And to our surprise, we found that across our 10 participants, there were really quite similar proportions of each one of these different cell types in our lungs. We also found that there were rare but consistently isolated monocytes or recruited-like macrophages in these healthy people, despite them having no pulmonary symptoms. We spent a long time looking at these cells and trying to figure out what they were. And what our best estimation is that these cells in blue represent immature monocytes that are maturing into macrophages, while this light green population is a novel cell that we didn't expect to find at all. In the paper, we called them matrix-associated macrophages because we didn't know what they were, but the genes that identified these cells were all genes that seemed to suggest they interacted with the extracellular matrix and might be involved in maybe homeostatic repair or interaction with the extracellular matrix. We now have come to appreciate that they really look very similar to macrophages in the interstitium of the lung, so macrophages within the parenchyma. This was very interesting to us. As I mentioned, this was kind of a novel finding, and we don't see a real obvious corollary in the bronchoalveolar lavage of a mouse. And so it was puzzling to us why we were seeing these in asymptomatic humans. And we reasoned in the paper that this must be some type of cell that either we are picking up the way we perform BAL in humans or some other cell that's unique to humans. And so from this study, what our main conclusions were is that to our surprise, we don't see a whole lot of heterogeneity in the macrophage pool if you take a random cohort of people from at least from the Denver metro area, and that although we can identify recruited macrophages, the proportions of them really are quite similar and quite conserved across 10 people, at least as far as we can detect using single-cell RNA sequencing. As you can tell by the date that this paper was published in 2021, we were wrapping up this project at about the same time that COVID happened. And so we were quite proud that we were one of the first people to publish single-cell RNA sequencing of human BAL, but boy, were we not the last. Within a year, I didn't count, but there must have been 100 papers with single-cell RNA sequencing of the BAL of patients with COVID of different severities or from people with acute respiratory failure for other reasons. And we looked at this data with great interest. we were really interested to find that in the majority of these studies, investigators identified a so-called pathologic or pro-fibrotic macrophage. And there was a lot of heavy informatics to try to understand what this pathologic cell was and what it was doing in the COVID lung. We saw lots of plots similar to what I showed, where there are some recruited macrophages, there are some resident macrophages, and then there's this mystery population in the middle that seemed to be increased in patients who had severe COVID. And there's a lot of plots like this that all of us looked at and didn't know what to make of it. But what we found is that a lot of these markers that were marking this pathologic macrophage were very similar to the ones that we called matrix-associated or interstitial macrophages in healthy people who had no symptoms. And they were found at a little higher proportion in those who had COVID, but their transcriptional programming looked almost identical. A couple years after COVID, this paper from some of our colleagues in Washington also came out looking at patients with ARDS. Most of the subjects in this study had ARDS associated with trauma, so not even an infectious etiology. And they too found this subset of pathologic macrophages that had many of the same markers that we saw in healthy subjects, albeit at a smaller proportion. And they showed in their study that the presence of this macrophage subset was associated with your risk of death, and although not statistically significant, a trend towards fewer ventilator-free days. We looked back at the literature too and looked at single cell from explanted lungs of people with other pathologies, for instance, idiopathic pulmonary fibrosis. And again, we found high frequency of these markers of macrophages that we saw as these interstitial-like cells in healthy people. And so I think in the field in the general, there was this hypothesis that there's specific pathologic macrophages that are driving fibrosis. But this didn't sit well with us because we thought if these are causative cells, why would they be in the lungs of people that are perfectly healthy and have no reason to need fibrotic repair in their lungs? And we thought perhaps the best way to get at the causation of these cells was to compare these findings with a normal type of lung repair. So not a fibrotic or pathologic injury, but maybe a sterile lung injury that we know resolves spontaneously and leaves no scarring or fibrosis. And so we developed a protocol based on one that has been used at the NIH since the early 1990s, in which LPS, bacterial endotoxin, which is purified, and you have to obtain it from the NIH to ensure its potency and quality, is delivered through the bronchoscope to healthy participants, and in a very localized way causes inflammation that lasts about 12 hours and then resolves spontaneously. Prior researchers have used this protocol and have shown that although it induces significant differences in temperature, heart rate, and gas exchange, these are considered clinically insignificant, and participants tolerate this protocol quite well. Prior publications had shown that using this protocol, you could induce neutrophilic inflammation that again peaks at about 24 hours after instillation, and by 2 days, neutrophils are gone from the lung. And during the same time course, macrophages increase. And we thought perhaps we could prolong this protocol or extend it and look at the macrophages that are present during repair from this lung injury and compare those to all of those single cell data sets that were published from COVID and other pathologic conditions. And so a few years ago now, we started this human endobronchial endotoxin challenge protocol. And just to walk you through it, our healthy subjects are screened to ensure that they're healthy and not at increased risk to undergo 2 bronchoscopies. In the first bronchoscopy, we lavage their lingula to obtain cells that are representative of the healthy lung. And then during that same bronchoscopy, we put the bronchoscope in the right middle lobe and instill endobronchial endotoxin into the medialmost segment of the right middle lobe. We then pre-assign these patients to come back anywhere between 1 to 5 days later, and we lavage that LPS exposed part of the lung and obtain those cells for our research purposes. We know from this protocol that this does induce acute lung injury. We know that from measuring albumin and total protein in the BAL fluid, and we see again that peak injury occurs at about 24 hours after exposure to this LPS. And by day 5, which is the duration of this protocol, we see that the lung barrier has been repaired as noted by the total protein and albumin levels going back to pre-exposure levels. If we look at the cell counts of the lavage fluid, we also see that neutrophils peak at about 24 hours after exposure and are gone by 2 to 3 days later, and that macrophage persist in the lung throughout this time course. And so to date, we've had 16 people complete this LPS protocol, and I'm going to show you single-cell RNA sequencing from the first 15. We had 4 people complete the protocol at 24 hours because we thought there might be the most variation there, and we have 2 to 3 people at each of the other time points. We depleted neutrophils from the BAL fluid because they are the majority of cells at 24 hours. And then we submitted all of these 30 samples for single-cell RNA sequencing. And we found some interesting results. So first of all, as we'd expect, the majority of cells in the lavage are macrophages, if you don't count the neutrophils. And across our time course, the percentage of macrophages really doesn't change all that much. There is a small proportional decrease as lymphocytes and dendritic and other inflammatory cells enter the lung. But if you recluster just those macrophages and look at heterogeneity within that group, you see an interesting pattern. Oops. So as one would expect, we see an influx of recruited macrophages at 24 hours after exposure, or day 1, and those cells persist in the lung. They're shown here in these purple and red colors. Resident macrophages, of course, decrease in proportion to those cells. And if you look at all of the surface markers that distinguish each one of these cell populations, we can pull out at quite a granular level all of these different macrophages. I'm going to summarize by saying the vast majority of clusters are resident macrophages. Again, we knew this from health, that there are small differences between these cells, but for the most part, they have overlapping identities. And again, they decrease at day 1 and slowly increase throughout our time course. We identified a number of what we termed intermediate cells because there were both resident and recruited cells in these populations, but they were defined by significant genes for production of interferons, metal binding, cholesterol metabolism, et cetera. These have been described by us previously and other groups, and they are a minority of cells at all time points and very, very little between our different subjects. Interestingly, in the recruited populations, we again see these 2 different subsets. So cells that look like monocyte-associated cells, or that they look like they're maturing from monocytes, these increase very quickly after exposure to LPS and stay elevated throughout our time course. And then these cells that we're now calling interstitial macrophage-like alveolar macrophages, or IAM. And they also increase after LPS. And although there's some more variability between subjects, they're present at all time courses. And so the first thing we wanted to do is look at all these different cell types that we identified after inflammation and say, do any of them have the same markers that we're seeing in all of these other pathologic conditions? And what we see right away is, yes, these interstitial-like cells, especially one subcluster of them, 4, looks very much like the cells that have been identified in all of these other pathologic conditions, despite this being a normal repair process. We went back to the literature and downloaded the raw data from the single cell of many of these studies. Here I'm showing one study of IPF, one of severe COVID, and that ARDS study. And we compared the gene signatures of these interstitial-like cells in our study to all of the different subsets of macrophages that were identified in their study. And we showed the most pathologic macrophage identified in their study in the right. And what you can see is that there's strong enrichment, overlapping identities of these cells across these studies. And this work was done by Elizabeth Nguyen, who is another biostatistician at National Jewish who's worked closely with us on this project. And so it seems as though this interstitial-like macrophage is present in many disease subsets and present in normal repair. We have questions about where this cell is coming from. This is very hot off the presses and posted to BioRx just a couple weeks ago. This is from a group out of Northwestern who's now doing spatial transcriptomics from patients with systemic sclerosis-related lung disease, and they again find these interstitial cells, and based on spatial transcriptomics, they describe these as interstitial macrophages that are spilling into the alveolar space. And so again, regardless of disease pathology, it seems that this is a conserved cell type that's somehow getting into the air spaces, perhaps from damage to the tissues. We're very curious to know what these cells are doing and what functions it is that is leading to fibrosis. And so we're just now digging into the genes that define these cells. It's a lot of data and it's a lot of heat maps. So I'm going to summarize by saying we think that they have unique signatures in a few functional domains. One of the more interesting ones is in their growth factor production. Of course, we'd be looking at that if we're interested in fibrosis. And again, you can see a pretty clear pattern that resident macrophages, the clusters shown in green to the right, and those that are recruited, on the left, and those that are recruited on the right have pretty distinct gene signatures when it comes to growth factor production. This pattern holds up if you look at the receptors that respond to all these growth factors. Also holds up if you look at cell death. Again, we talked about if these recruited macrophages are depleted, perhaps fibrosis could be prevented. They have very different cell death machinery and so are likely being induced to undergo apoptosis after inflammation in a unique fashion. They perform phagocytosis with different machinery. And they make very different cytokines and chemokines. And so I think there's a lot more work to be done here in identifying exactly which of these pathways and which of these genes are leading to the different signatures that are associated with these cells, more to come. But what we're concluding from this study is that, first of all, there is macrophage heterogeneity during inflammation, but it's conserved, widely conserved across now 15 unique participants, both during health and after inflammation. And the global identities of these macrophages are quite persistent through health, inflammation, and repair, suggesting that the identity of the macrophages and perhaps their functions is predetermined before they come into the lung and not as influenced by the microenvironment as we previously thought. We do note that the proportions change after inflammation and repair, so it's certainly possible that there's some tipping point where if you have more of one cell and less of another, that may influence your likelihood to undergo one type of repair or another. We certainly weren't able to detect that in our studies, and that there are some features that change over time, but the majority of them are shared across different cell types. Again, we're very interested in to understand these two types of recruited macrophages. We know that these monocyte-derived cells derive from monocytes. We're not sure where these interstitial macrophages are coming from, but we think that they may hold the key to understanding the reparative process. And we know that they are found in abundance during many disease states, not just ARDS. Okay, so our future directions. We're very interested in identifying, again, like I said, these unique functions of the interstitial subtypes. And so we're now grant funded to study these cells ex vivo and in organotypic cultures, things like alveolar epithelial organoids, understanding how they may promote epithelial repair. We're interested in knowing if the gene markers that define these cells are just markers or if they encode proteins that are important functionally. And so we're going back to our transgenic mouse models to do mechanistic studies on several of these markers. We're interested in quantifying these cells in patients with ARDS in a longitudinal fashion to see how their proportions may influence outcomes. And we're excited to partner with the ARDS, Pneumonia, and Sepsis Consortium, which has PIs here on campus. They also have a mini BAL protocol as part of that study. So we're interested in obtaining lavage cells from patients in our ICUs. And then we're also interested in expanding our LPS protocol. We think this is a pretty unique way to study cells from healthy patients and understand what functions of macrophages and other immune cells may be specific to pathologic conditions and which may be part of normal response to sterile lung injury. And with that, I'm going to stop and thank many people who supported us to do these studies. We work with a great group at National Jewish Health, and they've been supportive throughout my career to date. I'm also very indebted to the Center for Genes, Environment, and Health, especially Camille Moore and Elizabeth Wynn, who have really supported the interpretation of all this data. We perform our bronchoscopies at the Minimally Invasive Diagnostics Center at National Jewish, and thank all the participants who have allowed us to give them pneumonia for a short period of time. We performed single-cell RNA sequencing here at the U, at the Genomics Core, and we worked very closely with the NIH to obtain this certified reference endotoxin. And then we have been fortunate to have funding for these studies, but really these studies were made possible by the Betcher Foundation, who paid for all of the otherwise very prohibitively expensive RNA sequencing. And we just wanted to plug our study. If you know anybody who wants to be paid to get a short little batch of pneumonia, you can scan this QR code and contact our research coordinator. We obtain the cells post LPS to support studies in our lab, and we have many collaborations that depend on it. So we pay you, and that's all I'll say. I'm happy to take any questions. I'm interested in your comments. Thank you.
Great. Thanks, Dr. Mould. We'll get started with questions.
This is great work, fascinating results that you have. I guess with your pneumonia model though, and you're looking at these interstitial like macrophages in a fairly acute setting. And so how do you think that that transcriptional signature and function of those cells is going to change in a more chronic setting such as fibrosis?
Yeah, that's a great question. And I think it's to be determined. It's been fascinating to us. I think when single cell came on the scene, we thought that we were going to be able to find so much variation between people that we wouldn't be able to make sense of it. And the fact that we find the same cell markers and the very similar proportions across all these different people that we perform this protocol on, and from study to study, institution to institution, was really a surprise. And so I think there's a lot to be learned from comparing these very large data sets. You know, another, I think, big omics project that could be done is to look across these data sets, but of course you're always hindered by batch effect and experimental setup and so forth. And so one of the things we're hoping to understand by taking these cells and studying them ex vivo is exactly what you pointed out, the stability of these phenotypes and how, you know, microenvironment can affect these cells, and perhaps how things can change in a longitudinal fashion. It's been surprising to us how similar the global transcriptome is in a lung that is obtained from a cadaver in a fibrotic host versus acute LPS-induced injury in a very healthy person. And so I can't answer the question yet, but I think if anything, we've been surprised by how similar those two cell types have been.
That was very nice, clearly presented. So does anyone have good data on how long in humans resident macrophages live, like weeks, months, years, forever? And second, in mice, if you prevent the recruited ones from coming in by genetics or Crelox or whatever, how do they deal with the virus infection in the first place, like influenza or SARS-CoV-2?
Yeah, great question. So the first part of the question, how long do macrophages last in the human? We don't know. The best we can answer is from transplant studies, where of course you can identify host versus donor macrophages, but those are also very artificial settings where patients are on our immunosuppressants and so forth. So we don't know the answer. In a mouse, we think within a month, most macrophages have turned over, but it depends certainly on how much environmental toxins there are and how the immune system is activated. I think there are some very interesting studies going on in primates right now that, this is not the question you asked, but I'm just going to take the opportunity to say it, that these interstitial macrophages also make it into the BAL in primates. So we don't see it in a mouse, but we do think we see it in primates. And so it's unclear how well the mouse represents the lung, the human lung in these preclinical models. And it may be that primates our better model, but it's unclear. I think the mouse lifespan is very short and their life in the lab is very sterile compared to us humans. And so all of these studies about resident macrophages persisting through the lifespan may not be relevant in humans, and the primate literature would suggest that it's not, that probably at some point our resident macrophages might all be turned over by recruited monocyte-derived cells, but I think the jury's still out. And the second part, what happens if you don't have recruited macrophages? It depends on the pathogen, and I can't quote what happens in all different pathogens. In some cases, you have worse outcomes, and in some, you have better outcomes. We do think, though, that you're protected from fibrosis.
Kara, great talk. What kind of intervention could you foresee that would preserve the normal immune response, which is obviously very important in the acute phase of the infection, and yet abrogate the maladaptive fibrotic response that you see, with your patient example causing fibrosis at 2 months?
Thank you for that question, Dr. Badish. I wish it was my idea, but it's not. There's currently a clinical trial going on for an anti-CD4, and I think it's a small molecule, might be an antibody, but the idea being that if you can stop the ongoing recruitment in pulmonary fibrosis, that perhaps you can halt fibrosis. I'm not sure that that will ever make it to the acute inflammation, because again, we think we need these recruited macrophages for normal repair. So when you give LPS, these cells come in and the lung goes back to normal. So I think you have to be able to identify the point where normal repair becomes exuberant repair and fibrosis, and I'm not sure we know how to do that yet. I also am not sure we know when repair starts. So it may start during inflammation, and I think it's hard to know the timing of those therapies and when they might be safe, but I know they are interested in that idea in patients with established fibrosis. So more to come.
So very, very nice talk, beautiful data. Ultimately what you're trying to do is get at, how do these interstitial macrophages drive fibrosis in different situations? And so there are many possibilities of how that might happen, but simplistically, one is that the different interstitial macrophages from different people respond differently to the insult, or that another is that cells around those macrophages are responding differently to what would otherwise be normal signaling and repair. And I know that it looks like you're getting at that in part by doing the organoid, cultures and probably co-cultures and things like that you're planning to do. But, I may have missed something that you said, but could you talk a little bit, do you have any data right now as, from interstitial macrophages from people that go on to get fibrosis versus those who don't from whatever cause?
We like to think we do, because we have lots of mini BALs banked from people with acute lung inflammation, but no, we have not done the study where we look at the interstitial macrophages from people who do or do not go on to develop fibrosis clinically. And as you can imagine, that's really confounded by the therapies, by a million different cofactors that are or confounders that are going to be hard to control for, which is why we thought the LPS protocol would be a way to get at it, because it's standardized. You can understand the baseline health, the medications everyone's getting, the time resolved kinetics of this macrophage programming. And they do change somewhat over time. And so I think that's going to be the next frontier, if you will, but it's going to be very challenging because of the heterogeneity of all of us humans in an ICU setting.
Kara, as a simplistic ID doctor and non-macrophage person, you made this incredibly accessible, so thank you. As you probably could guess, I have a clinical and a public health question, which may be combined. From a clinical perspective, are there, if I'm seeing someone in your ICU, if we're seeing someone in the clinic or in the wards, are there hints or markers from a clinical level that this person may have a more robust negative response. That's number one. Number two, from a population health perspective, are there environments or settings where we know that, or you know that that the maladaptive macrophage response is more likely to occur? I'm looking for modifiable environmental and clinical clues.
Yeah, thanks for that question. I think we all have many hypotheses about what those modifiable factors were, but I think it would be disingenuous to say we have strong evidence. So I don't want to overstate evidence currently. So I think age is one that many people have looked at. It does certainly seem like older age is more likely to be associated with fibrosis. And so there's a huge interest in the field in senescence, or this idea that older cells don't do as well. We think, again, that having more of these recruited macrophages, so having prior insults to the lung, whether that be through pollution or prior infection, et cetera, makes your lung less so-called resilient. Again, we could blame the macrophages, but it's much more likely to be a combination of factors and epithelial cells and so forth. And so there's a lot of work being done to measure previous exposures. So whether it be heavy metals or particulate matter, et cetera, and whether those patients go on to develop worse adaptation to acute lung injury versus those who have clean country living lungs. I think most of what we have are anecdotal or weak studies right now, but it's certainly a hot area of research in the field.
So that was a fantastic talk, Kara. Thank you for, and I think the thing that's so impressive about it is just seeing how your work has progressed since the time of your fellowship. So congratulations on all of that work. I'm going to ask a question that's a little bit upstream of the development of fibrosis in patients that have underlying lung injury. So Carolyn Calfee and others have shown that there are a variety of different endotypes associated with ARDS, you know, the pro-inflammatory type and then a non-inflammatory type. And so prior to the onset of fibrosis, do we have any knowledge of how these recruited macrophages are impacting the underlying endotype of ARDS and how it might inform kind of what we do with therapeutics earlier?
That's a great question. I hadn't actually thought about that, but yeah, it does beg the question. You know, we thought that perhaps the translational aspect of this research would be that we could look at monocytes, precursors in the lung, and be able to predict something about what the recruited macrophages are going to do. So in a way, it's kind of annoying that it's these interstitial cells that we can't easily get at that might be doing all of the damage or help or whatever, we don't know, all the repair. It would certainly be more convenient if we could find some signal in the monocytes, but we don't think that's going to be the case. Several of those mediators that Carolyn Calfee initially measured can be produced or affected by macrophages, and we know through some other work that local mediators that get produced in the lung during acute lung injury do show up in the circulation. And so it's certainly possible that whatever's going on in the reparative milieu of the lung could be detected in the circulation, and perhaps we'll find that one of those factors that Dr. Calfee has measured really is coming from the lung, but we don't know yet. I think it certainly is plausible to think that someone's immune endotype would affect this process. I think the APS consortium may be a good place to answer that question, since we'll have so many measurements, and certainly those ARDS endotypes are a focus of that study.
All right. Well, thank you so much, Dr. Mould, for that.